First of today, I am deliver the lecture on the topic of the microorganism antagonistic to the plant pathogen in soil, rhizosphere, and phyllosphere, and their management of the plant diseases. First of all, see this image is correctly the fungus grow in the fatty plate. Another is that two different plate here. See the first of all. This is the two amendment medium. One is the first, which is the glycerin and the growth inhibition of the Rhizoctonia solani, causing the seed blight of the rice by using the trichoderma virens, the another antagonistic microorganism. They produce the antibiotic just like the glycerin. The first amendment is the glycerin, the amendment medium, and another is the no any amendment medium is the inhibition of the Rhizoctonia solani mycelium growth is. First of brief all the contents and introduction. The first of all antagonistic microorganism. First of all, the H.S. Smith in the 1919, the first term they use the biological control or biological control, the use of living organism to suppress the population of a specific pest organism, making it less abundant, less damaging than it called the otherwise. Biological control, the use of microbial antagonistic including bacteria, fungus, virus, nematodes, and other to suppress the plant disease pathogen. And the first of the Paul DH in 1964, the differentiate the natural control and form the biological. Two are different terms used in the agricultural natural control and biological. Both are different. Natural control, that means the maintenance of a more or less fluctuating population density of an organism within certain deep level upward and the lower limit of a period of time by action of the abiotic and biotic environmental factors. There is a figure the penetration of the host area of the formation with the larger hyper of the Rhizoctonia solani by the smaller hyper of the Trichoderma virens. Why biological control is that today is the most important because so many highly highly toxic substances are used for the disease management, but these are contaminated the environmental agro ecosystem. So now time they move to the agrochemical to the biopesticide. They are playing the why biological control are used. They are highly economically selective with no side effects, self propagating and self multiplying. खुद से ये मल्टीप्लाई करते हैं, खुद से ये propagating करते हैं. Pest resistance to the biological control agent is the virtual known, and the no harmful effect on the human, livestock, and other organisms. The virtually to the efficiency, greater ability to search their area, the improve the quality of the produce compatible with the most of the IPM component. They are following characteristics. The what biological control characteristic. The importance of the biological control. Why the importance of biological? Why to move the we are moved to the biological control? क्या क्यों हम biological control की तरह move कर रहे हैं? क्या इसका reason है? What should be reason? There are so many reasons that available in the long term. Biological control are the very most effective in the long term effective, cost effective, but safe to for human. Safe for the agro ecosystem as well as environmental condition. The alternative method of the disease control because to substitute the high toxic substances, the non-toxic to man, animal, birds, environment act as a selective target organism. They are only one organism they target to the another organism, just like Trichoderma virens or Pseudomonas vibrio bacillus. Another species are used to the invade the another plant pathogenic bacteria and fungus and viruses and the cross protection also. Reduce the possibility of the including resistance in the pathogen. It is self-sustaining and easy adaptation to the any environmental condition. Limitation of the biological control. There are so many limitations, but slow action. They are slow action. They're expensive, difficult to develop, labor expensive, highly specificity to the host, and the pathogen, strain, biotypes. The highly selectivity, a skilled person are required to the handling of this biological control. Do not just like a normal the handling the biological control. A skilled person, highly skilled person are required to deliver. But these are after the training period of the man is the also handling of the biological agents. Variability is the effectiveness is more susceptible to environmental conditions, short shelf life of the formulation, storage problem. There are so many limitations, but they are after the known there are no limitation effect problem. Chemical pesticide none the biopesticide. Why do use the chemical pesticide? Chemical pesticide the polluting of the toxicant resistance to phenomena effectiveness does not depend on the biotic and abiotic factor control the industrial production. But in case of the biological control, genetically modified organism, natural products, microorganism, non-polluting, the non-toxic substances, they produce and the multiple mode of the action they inhibit the growth and they also enhance the growth of the plants and increase the yields and economic importance. The effectiveness depend on the biotic, abiotic factors, complex industrial productions. How to increase the efficiency and improve the biological control? There are so many mechanisms available in this. Yes, sir. Yes, sir. I am audible. Sir,
yes, you are. Yes, you are. Yes.
Okay, sir. There are so many mechanisms. The mechanism of the microorganism which the antagonistic microgen affect the pathogen population are not always clear, but they are generally attributed to one of the four effects. First of the direct parasitism, या lysis, या death of the pathogen. ये पहले parasitize करते हैं या lysis को करते हैं, then after death. Yeah, another is the competition with the pathogen by the antibiotic substances released by the antagonistic, and the last one is the indirect toxic effect on the pathogen by volatile substances such as the ethylene that is released by the metabolic activities of the antagonistic. How to increase the efficiency and improve the biological control? They are so that the efficiency of biological control should be, I think, improved for desirable characters, which result in the better management of the plant disease. Just like mutation having, they are the protoplast fusion, genetic modification, and transformation. These are all methods are also used to improve the efficiency of the biological controls. The mode of action of the biological control: antibiotic mediated suppression. The antibiotic are the microbial toxin that can low concentration poisons kill to another microorganism. Biocontrol agent and their mechanism is available in the plant disease management, inhibition of the pathogen by the biocontrol bacteria and fungus, and the antibiotic mediated suppression. The antibiotic are microbial toxin that can at low concentration kill the other microorganisms. There are two types: the volatile and non-volatile antibiotics. Volatile antibiotics, that means hydrogen, aldehyde, alcohols, ketones, sulfides, and non-volatile including the polyketides, the damping and the meplicon, and the heterocyclic nitrogenous compounds. Parasitism, they are two types of parasitism: the direct utilization of the pathogen as a source of nutrients, just like the microparasitism refers to association in which a parasitic fungus, like the parasitic another fungus, and also is known as a hyperparasitism when a hyperparasite biocontrol fungi and utilize the hyperparasite pathogenic fungi as a source of nutrient. The hyperparasite through the parasitizing hive to acquire the host nutrient may also require the cell wall degrading enzyme for the complete parasite. There are so many steps are involved in the microparasitism. First of all, the chemotrophic growth, recognized stage, and attachment to the cell. The first chemotherapy growth, the by control of the fungi grow towards the target fungi, chemical stimuli, and another is the recognition stage, the interaction with the biological control receptors that of the host fungus. Another is the attachment of the cell wall degradation, just like the chitinase and the glutinase, and penetration, just like the penetration, the procedure in the case of the fungus, the procedure like structure to penetrate the host cell, they do not. There are so many litigation, just like biological control, the serial action, the exploration of side disease, the mechanism of the chitinase, and the references were originally 1998 examples. And amendment of the plant growth substitution with the chitosan, the action in case of the Fusarium oxysporum, the radicals glycosid, their mechanism the chitosan. In this figure, there are two represent the figure. The microparasitism of the Rajactonia solani by the trichoderma virus. A figure A, they represent the parent strain coiling around the host hypha, just like coiling the host hypha, and another is the microparasitic deficient, the mutant with no coiling, any penetration of the hypha. In this coiling, just like the microparasitic, just like trichoderma virus is the coiling in the host and the parasite, and another is the really the microparasitic deficient mutant with no coiling and no penetration of the hypha. Competition, the competitive exclusion of the pathogen by biological control agent via just like the competition for the SPS nutrient, etc. And the production of substances such as the cytophores for the nutrient, such as the iron's acquisition, and the deprive the pathogen for the nutrient. It is an indirect mechanism of competition for SPS and the nutrient between the Pythium irregular pathogens, the white restricted growth, and the Epicoccus purpurissum, the reddish white growth on the PDA medium afterwards. In the soil microorganisms compete for the substrate and the infection site, just like biological control agent are used to the essential element like the carbon, nitrogen, and any other influence, just like you make unable to the pathogens. And example, just like example, quoted in the potato escape and the bean root rot by the green manuring Fusarium species, just like the Fusarium is a very highly destructive pathogen. They cause so many economic important disease, and losses the high amount of the economic yield in the case of the banana and any other wild seed and grain. Fusarium and Pythium species why the other soil and plant associate with the microbes. Pythium ultimatum by the Enterobacter clazy through the effective catabolism of the nutrient in the spermosphere. Cytophore production of the plant growth promoted as a bacteria, just like Pseudomonas fluorescens control the Erwinia carotovora. This bacteria is the control the Erwinia carotovora. The Erwinia carotovora is a destructive pathogen because they can induce systematic resistance. Also known as systemic ICAR, systemic resistance in the plant variety, the pathogen induced by the presence product of the biological control. And the necrogenic pathogen, that's like the necrotic pathogen applied on the leaves, non-pathogenic bacteria, just like applied to the root seeds, the metabolites of the pathogenic saprophytic bacteria. And another is the metabolites able to induce the ICAR, just like polyacrylamide, elixir, thylene, salicylic acid, acetyl cyclic acid, amino acid, harpine, Erwinia, and already harpine is developed by Erwinia and and the stress can induce defense mechanism against a pathogen, just like physical thickening of the cell wall, the cell lignification, deposition of the calor, and accumulation of the antimicrobial low molecular weight substance. And the just like phytolagins is the low molecular substance that also help in the plant defense system, and synthesis of the various proteins, just like chitinate, gluconate, and other pathogen protein. These are also the produce the defense, help in the defense of the plant disease against to the plant pathogens, and ICAR defense response. In this, a plant respond to a variety of the chemical stimuli produced by biological control agent, just like so many biological control like previously discussed, such as the stimulant induce the defense in the plant. The first of the pathway called systemic acquired resistance, and another the second pathway is mediated the salicylic acid. In the second case, the referred to as the induced systematic resistance is the mediated by jasmonic acid and the ethylene. The single transaction pathway the leading to the pathogen, the single pathway leading to the pathogen, they induce the systemic acquired resistance, and the rajibacteria mediated a ICAR in the Arabidopsis thaliana. There are so many bionucleatics of the different types of enzyme, just like the antibiotics, hyperparasitic enzyme, chitinase enzymes, production of the lactic enzyme, ICAR, cytopol. These are all enzymes that are produced by the bionucleant agents. Another is the hyperparasitic the predation, just like the obligate bacterial pathogens. Hyperparasitic facultative parasite predation, just like in case of the target organism, just like the biological control agent, the gastric penetrance, the action just a root not nematode, the hypovirulence, the virus is the cryptorachia, the parasite kind, the facultative parasite, the cladosporium, oxysporum, the cladosporium, the viruses. In the case of the powdery mildew fungi and the predation, the trichoderma species, there are the just like this is described in the first slides. Another mechanism of the biological control, they are the three mainly mechanisms, the types of the direct antagonism, mixed path antagonism, and indirect antagonism. Antagonism. The direct antagonism, the hyperparasitism is a predation, just like example of the species, the trichoderma species, and in the trichoderma species, and mixed path the antagonism, and just like the antibiotics release करते हैं, lytic enzyme release करते हैं, unregulated waste product release करते हैं, physical and chemical interference. In case the salicylic lipopeptides, chitinase, ammonia, and the pathology of the soil spore. In case of the indirect antagonism, the competition and just like a mechanism, two types of mechanism, the competition and induction of the host plant resistance. In the example, just like cytopol SKV and the contact with the fungal cell wall, and another the Phytophthora mediated. In some example are available here, the biological control agents and against pathogen used, just like in case of the fungi, the Trichoderma harginium, and used the genus for Gramineaceae tetsi and Asclepium rolfi, Fusarium oxysporum prevents, and the Trichoderma viridi, just like it was used the Nivosia indica, Nivosia indica is very destructive. So these all pathogens are very destructive. They cause the economic ill. Just like in case of the bacteria, the Pseudomonas fluorescens subtilis, these two are used in the particularly. The Pseudomonas fluorescens are used in case the against the Pyrifloria oryza and the Rhizoctonia solani and Asclepium rolfi, and another is the Bacillus subtilis are used against the Fusarium odum. They cause the gluten, and virus is the Bacterovirus, and the another is the nematodes, the Leptospirosis lineage E. coli. There are so many antibiotic mediated suppression. The microbial toxin that can at the low concentration poison. The antibiotic just like the agosine, 18-4, 2,4-deacetylcysteine, eleptoxin, mycostinizin, gentosin. These are sources. They are the agrocin are the isolated from the agrobacterium radiobacteria, and the used for the target pathogen agrobacterium tumefaciens. They cause the crown gall root of the roots in the plants. And another 2,4-diacetyl plugins, they sources isolated from the Pseudomonas fluorescens strain F1 and 3, and the target to the against the PKM species, the very destructive pathogen, and they were maintained in the very low moisture condition in acidic soils, and they causing the damping of diseases. Regular toxin is the isolated from the sources the Trichoderma virens, and the target pathogen the Rhizoctonia solani, they cause the syphilitic rice, root rots, both are the microstimulants. Another is microbulation, and microbulation is isolated from the Bacillus subtilis BBG 100, and there is a used against the PKM, where there are so many antibiotics and isolated source and used for the target pathogen and the causes the disease, and the with references in this slide mentioned. Another is the bacterial influence, the bacterial strain, just like Bacillus the microbial strain बैक जी बोलते हैं. आई थिंक प्रीवियस ईयर में पूछा भी गया था इस माने दो एक कंट्रोल एजेंट का असरफ में यहाँ से क्वेश्चन पूछा गया था. इन केस ऑफ द सुगर बीट द बैक्टीरियल determinant pyroxide chitinase and beta-1,3 glucans gene are responsible for the ISR induced systematic resistance.
Hello, sir.
हाँ, hello. आप लोग थोड़ा हमें बताइए व्हाट इज द SAR एंड ISR, व्हाट इज डिफरेंस बिटवीन द SAR एंड SNR, which types of molecules ये which type of molecules produced by SAR in case of SAR and ICAR? Anyone explain to me? Okay, Pune. Just like a Bacillus subtilis, the GB03 and the IN937 are the plant species the Arabidopsis, and the bacterial determinant 2423 butanediol in the ISR systems in this systematic resistance, and the Pseudomonas fluorescent strain is the CHA0 in case of the plant species tobacco, and the WCS 417 carnation, radish, and tomato. Just like a bacterial determinant, the cytophore and the lipopolysaccharides, and just like induce systematic resistance, and in case of the tobacco cytophore, the systematic acquired resistance. Another just seed the seedling root tips in transplanting crops, the foliar sprayer, soil drenching, pronized along with the compost, farmer manure, vermicompost recommended for the plant, the by control agent, trichoderma, especially they are so many figures they explain in the each two steps, and the 3 to 5 gram per kg of the seed for the seed treatment, and the 8 to 10 gram per liter of the water for the seedling dip, root dip, soil drenching, and foliar sprayer. This is recommended by the blood it all in 2001. I'm audible?
Yes, sir, you are audible.
Sir, I am audible?
You are speaking too fast, sir.
Yeah?
Yes, sir, but you are speaking very fast, sir.
Okay, sorry. Just like it first is the slide, the plant growth promoting rhizobacteria, and several plant growth promoting rhizobacteria are available, and they are used for the enhancement control to the plant pathogens, especially the root rot pathogens in the several plant bacteria are the currently used, the commercially available also, and they are called different name and operate through the range of the mechanism. First of all, the bioproteinants through the suppression of the plant disease, and another is biofertilizer through the improve the nutrition acquisition. जब बायोफर्टिलाइजर का अगर यूज हम लोग करते हैं तो वो न्यूट्रिएंट एक्विजिशन को बढ़ाता है, they increase the they improve the plant health system, and they also जब plant का improve health system अच्छे हो तो वो डिफेंस काम करेंगे, they escape कर सकते हैं पैथोजन को. Another is the biostimulants. Through the phytohormone production, through the phytohormone, the hormone produced by the plant, and which are also act as a defense system against the plant pathogens. Plant growth promoting rhizobacteria, the bacteria, especially it is colonize the plant roots. The plant roots throwing the inoculation on the seed, and that enhance the plant growth system. They are flowing characteristic of the plant growth protein that they are because the must be the proficient to colonize to the root surface, and another is the must survive, multiply, compete with the another microorganism, and another is the must promote to the plant growth promoting bacteria. Plant growth promoting rhizobacteria promote the plant growth by two means, the direct indirect means. Direct means the direct mechanism. The indirect mechanism enhance the plant growth in the absence of pathogens. The pathogen नहीं होगा तो उसको plant के growth को enhance करेगा. जब कोई भी plant अगर escape कर रहा है, pathogen उसको नहीं है attack किया, कोई भी fungal किया, bacteria any pathogen का attack नहीं है, तो वो ग्रोथ को प्लांट की ग्रोथ को इंप्रूव करेंगे. एंड अनदर इज अगर वो इनडायरेक्ट का मतलब इनडायरेक्ट दैट मींस कि अगर अटैक है तो वो उसको एस्केप करेंगे. It can be categorized in the following groups, just like plant growth rhizobacteria, the nitrogen fixation, phosphorus utilization, cytophore production, modulation of the in case of the I think the phytohormone levels. In case of the nitrogen fixation, there are a number of plant growth promoting rhizobacteria are available, and which are enhance the nitrogen fixation in the crop, which are able to fix atmospheric nitrogen and make it more susceptible to the plants. Another example, just like example आप recall कीजिए आप लोग, जैसे rhizobium is the common bacteria and the root nodules bacteria. They form the nodules on the root of the leguminous plant, just like the chickpea, lentil leaves, such as a soybean, peanut, alfalfa, chickpea. Also and azosporidium, another is the azosporidium is a free living nitrogen fixing bacteria that can be fertilized with the wheat, sorghum, etc. Another is the categorization, the phosphate solubilization. The low level of, in case of the low level of the phosphate can limit the growth of the plant as it is not the available to growth of the plant. Some of the plant growth bacteria that solubilize the phosphate from the either organic or inorganic abundant phosphates, which is facilitated to the increase the plant growth system, and also the act as a defense to against the plant pathogen bacteria and abiotic factors also. Another is the just like the H. species and Cyanobacter, there are two species are available and which enhance the nitrogen fixation in the leguminous crops and phosphate solubilizing. Two bacteria are the phosphate solubilizing types of bacteria, and another is the cytophores. Iron is a vital nutrient for the almost all from the life, and despite the abundance in the particularly in the soil, natural availability of the relative low, the many microorganisms possess highly affinity iron uptake system mediated by the action of the low molecular weight iron chelated. Another is the phytohormone, Pseudomonas fluorescens is the bacteria which involve the cytophore production in the leguminous crops, and another is the mechanism, another fixation categorized as a modulating phytohormone level bacteria. The modulating phytohormone level bacteria is an example of the Bacillus species, Pseudomonas, Rhizobium. These all bacteria are at the phytohormone levels bacteria. They produce phytohormone in the plant system. The phytohormone play a key role in the plant growth and develop and respond to the adverse environmental condition. Under the stress condition, the plant attempt to adjust the level of the indigenous plant hormone in order to decrease the negative decrease the negative effect of the environmental stress condition. Under the such condition, rhizosphere microorganism, a modulating phytohormone level bacteria, promoting the plant growth. Azotobacter crococan is the Javelin catenate indole acetic acid are produced, and these all are the just like Bacillus already described, the Bacillus and Pseudomonas rhizobium. These all bacteria are the phytohormone bacteria. They are the plant growth promoting bacteria are the indirect mechanism involved. Indirect, that means indirectly by the decreasing the inhibitory effects of various pathogens on the plant and development of the in the form of the biological agents. Just like example, the antibiotic, lytic agent, they produce indirect means the antibiotic and lytic agent, the production of one or more antibiotic is the mechanism most commonly, associated with the ability of the plant growth promoting bacteria and antagonistic agent to phytohormone. Another is the previously described the competition. The plant will compete with the pathogen for the nutrient, and is the rhizosphere and Pseudomonas fluorescens PCL. Pseudomonas extraneous Pseudomonas fluorescens suggested that the control the disease, the Fusarium oxysporium in the radicals. There are so many literature are available. I think in the case study, the integration between the compost, Trichoderma hargenium and essential oil for the controlling the peanut crown nut root under the field condition. This paper is the main objective, the potentiality assessment of Trichoderma hargenium and it the combination with the compost essential oil for controlling the peanut crown nut disease. इस पेपर का मेन एम ये है कि इसको हमको चेक कर रहे हैं, इन्होंने चेक किया है कि ट्राइकोडर्मा हार्गेनियम का क्या एफिशिएंसी है कंपोस्ट के साथ, एसेंशियल ऑयल के साथ. Essential oil, so many essential oil are available, the volatile oil, but they are usually so many restricted, and very few essential oil are used for the plant disease management nowadays. just like citronella. Citronella oil is the best oil. I think in case of the brown spot of rice for the management bipolar sporadium. In this table, the different essential oil concentration, the essential oil, the thyme, growth, and lemongrass, and the control, the concentration 0.25, the thyme 0.2551. The three concentration of the each essential oil, thyme, growth, and lemongrass, and control is no any other area. And the pathogen, linear growth and reduction. Linear growth, the highest growth, thyme, essentially when you use the thyme, the concentration to 0.25%, the linear growth of the pathogen 31.0, and reduction was 65.5. That means when you use the low concentration of thyme in essential oil, the highest linear growth absorbed in the 0.25 at level of the 0.25, and reduction, low level of reduction of the aspergillus niger. In case of the highest lemongrass oil, the conclude that the thymine at the concentration 0.5 and 1%, 0.5 and 1%, the 100% inhibition of the linear growth and the reduction of the reduction. Another the table is the difference, the effect of the combined treatment with the biocompost and essential oil and the pea yield under the field condition. Just like example, the trichoderma compost, having oil, primrose, lemongrass oil, single treatment, and the increases this efficiency. The highest efficiency increase the trichoderma hardynia with increase the 25% yield. And other companies, the compost plus trichoderma, hardynia plus thymine. And the increased efficiency highest is compost blood trichoderma hardynia and the lemongrass oil, the highest efficiency and the increase the 5.7% under field condition. Another case study to check the ability of biocontrol agent Pseudomonas syringae strain to control the bacterial escape to the tomato. In this table, there are so many biocontrol agents, just like this idea, the light brand A506 Pseudomonas fluorescens, A506 Pseudomonas syringae, there are so many strain, B41, the B52, and the foliar incidence. There are two parameters are observed in the foliar severity and foliar incidence. In this figure, the lowest foliar incidence was observed in case of Pseudomonas syringae and the CIT7. This is the finding of this paper. The biological control involve the use of microbial antagonists such as the bacteria, fungi to suppress the plant disease pathogen, and the biocontrol have the several importance in nowadays and the very important agent for the survival for the defect to the plant pathogens and advantage of our control methods. There, the mode of action include antibiotics, competition, parasitic, induced systemic resistance. They are also previously described. There are, however, some limitation at the generally use of the biological control agents and variability of the effectiveness of and low spectrum action. I think. You have explained any the SAR and ICAR in case of biological control. Biological control agent can be applied as a seed treatment, root dip, soil, furrow drain, foliar spray, yeah, through the drip irrigation. There are so many the methods of the use of the biological control. या root dip भी कर सकते हैं, seed treatment कर सकते हैं, उसको furrow drenching के लिए यूज कर सकते हैं, या foliar spray के लिए भी हम लोग यूज कर सकते हैं. अभी तो सारे एक जो भी कंपोस्ट है वो 3 methods में है, and another जो through the drip irrigation के through भी हम लोग biological control का application कर सकते हैं. And devise better the strategy for the screening of the biological control and develop the upper super strain with the augmented biocontrol efficiency. Another is the development of the microbial construction. Another is the compatibility to check the competitive biological control agents, the different type of the agrochemicals available in the market. Then another improve the improve the knowledge on the efficacy related issue, and another they promote the multidisciplinary approach to integrate better to the IPM approach and the safeguard the durability of the biocontrol agents. Training and advice to the farmer to the development and dissemination of the disease support system to DSS को उसको हमको support लेना पड़ेगा. Another is the establishment of the demonstration scheme. Demonstration को हमको enhancement करना पड़ेगा ताकि किसी भी product का demonstration हो और farmers ज़्यादा से ज़्यादा use करे उसको. Then after जो उसका जो feedback आएगा उस feedback के आधार पर हम लोग उसको improve करेंगे. अगर कोई drawback आता है उसमें कुछ limitation आता है तो उस limitation को बाद में दूर किया जाता है. 1 minute. Another topic is the important cultural practices and their role in the plant disease management, control, solarization, and in IDM systems. Slide is visible. Sir, slide is visible. Hello, sir. Slide is visible. Hello, sir. Why adopted the cultural practices? First of all, the plant protection in India and the most of the developing country is mainly based on the use of pesticidal chemicals. And chemical control is one of the effective and quicker methods nowadays in the reducing the pest population, where the farmer get expected result within the short time. Farmers का ये need होता है कि जल्दी बहुत short time period में हमको result आए. That means कि देखो कोई भी chemical spray कर दे but resultant जो positive हो और short time में हो. इसके लिए farmers chemical control की तरफ previously move किए थे, but now time this is escape. However, जब over the religions and the indiscriminate use of pesticide resulted in a series of problems in the agricultural ecosystem, mainly the development of resistance to insecticide, resilience of the treated population, outbreak of secondary pests into the primary nature, environmental contamination, residue hazards, destruction of natural image of the insect pest, expenses on the pesticide equipment and labor. Dear student, I am audible. First of all, why adopt the cultural practice? Cultural practice हम लोग क्यों adopt करना पड़ा? क्या इसका reason था? क्या drawback हुआ जो हमको cultural practice के तरफ move करना पड़ा? First of all, the plant protection in India and the most of the developing country is mainly based on the use of pesticide chemicals. And the chemical control is one of the effective and quicker method in the reducing the pest population, where the farmer get expected result within the short period. हमारे farmers का ये need होता है. कहीं भी आप चले जाइए, किसी भी country में चले जाइए या माने या state में चले जाइए, किसी भी village में चले जाइए, आप वहाँ का जो farmers मिलेंगे आपको एकदम different मिलेंगे, because कि उनको quicker result बहुत जल्दी result चाहिए, एकदम short period में result चाहिए. और शॉर्ट पीरियड का रिजल्ट केमिकल पेस्टिसाइड हो, बायो पेस्टिसाइड हो, इन अदर मेथड्स हो, कुछ भी हो, लेकिन उनको रिजल्ट चाहिए. उस कारण से प्रीवियसली द केमिकल कंट्रोल की तरफ मूव करे। इसका रीजन एक और भी है कि प्रोडक्शन प्रीवियसली बहुत कम होते थे, वैरायटी माने हाई यील्डिंग वैरायटी आर नॉट अवेलेबल previously, so they can defend to the chemical control. And the another, the reliance, the indiscriminate use of pesticide resulted in a series of problems in the agricultural ecosystem, mainly the development of the resistance in the insect to insecticide, and another contamination, residual hazard production, and the natural enemies, insect and destruction of natural enemies, and expensive the pesticide and equipment and labors. All these problems accumulated and contribute to a new way of the thinking, considering the pest control practices as just like the integrated approach of the pest control. The integrated pest management is the ecological approach methods. Is the ecological approach in which utilization of all available techniques of the pest control to reduce and maintain the pest population at below the economic injury level. आप बता सकते हैं इकोनॉमिक इंजरी लेवल क्या होता है? Anyone can explain the economic injury level? Hence the new concept of approach is based on the principles of the managing the pest rather than the eradicating them. Another word is the pest management will be the working 25 hours a day, every day of a year. This is possible to the must become the part of cultivated and the thinking activities. Disease, the American Pathological Society, according to American Pathological Society, the disease is a malfunctioning process that is caused by the continuous irritation, which result in the some suffering producing symptoms. The plant, therefore, the plant disease is a structural abnormality, physiological disorder. Plant में कोई structural abnormality होता है, कोई भी किसी प्रकार का disorder अगर दिखाई देता है, both due to an organism या unfavorable condition. ये दोनों किस लिए होता है? या तो कोई organism का attack हो या otherwise unfavorable condition हो. That means कि so many unfavorable conditions including the low level of nutrients and other microorganisms, environmental pH, temperature, moisture, and relative humidity. They can affect the plant या its part या product may reduce their economic value. Integrated disease management is a concept derived from the successful integrated pest management system, which consists of according with the timely application of combination of the strategy and tactics. आइडिया में एक practice एक approach हमको provide करते हैं जो कि हम लोग timely उसको management किसी भी disease को या किसी भी practice को ये timely हम लोग use करके उसको management कर सकते हैं. There are so many importance of the plant disease. The plant disease is sometimes spread as a epiphytotic and destroy to the crop growing in the very large area. जैसे आप लोग जानते हैं boom and bust area वहाँ पर जो vertical gene का जो विषय होता है जिस कारण से boom and bust वो तो theory आया था. They damage to the crop growing in the field as well as store product in the storage, and this disease can occur any time and any stage of the plant growth, and the sowing seeds, storage product, and the cause the great economic losses. In Asia alone, the food grain amounting millions of the dollars are being destroyed every year due to the crop disease. एशिया में देख ही रहे हैं मैंने अभी इस चीज को ऑब्जर्व कर रहे होंगे. The example of the sum of the epiphytotics, the late blight of potato by fungus Phytophthora infestans. This Phytophthora infestans is a very destructive pathogen. They cause the high amount of the late blight and the starvation in the Ireland country, and then another also in the India. Coffee rust, the Amelia vastic in the Sri Lanka in 1817, and cicatrica leaf disease of the banana and caused by Mycosphaeral amicicala in the Central and South America. The cicatrica leaf, the cicatrica leaf spot disease is the very destructive disease in the Bihar climate regions. They suddenly within 1 week they spread this pathogen and throughout the field, and a colonization of the host, and they produce symptoms, and a great amount of the symptoms go in the plant. They destroy the plant, and the very quick fasting of the pathogens because moved by the air. This is airborne pathogen. Cicatrica, Mycosphaeral amicicala is airborne pathogen. Another is the India. In India, the famous Bengal famine 1942 was due to the early spot disease of rice, just like brown spot disease of rice. called by bipholates or helminthosporiaceae, and approximately 2 million people died of the extrovasation. Plant disease is caused by a variety of causal agents, just like fungi, bacteria, viruses, phytoplasma, nematodes, and another pathogen just like parasitism. And the another crop can be attacked at the different growth stage, at the seedling, stem, root, seeds. Beginning in the early 18th century, the first bring of the grain with the salt water, the salt water used for in the 17th century, and they used for the control of one disease of fruits. And another the pre-host research in the 1807, the copper sulfate. Copper sulfate are also demonstrated in the first time for the seed treatment against the one disease, and Millardet is the very in 1822, they discovered the vortex moisture to the control of the downy mildew of the grapevine, because the grapevine very downy mildew is the very destructive pathogen in case of the grapevine disease. They destroy the orchard of the grapevine, and also destroy and the lack of the wine industry is also economic loss in the wine industry. We developed the chestnut compound in the 1921, and Sanford in 1926. The antagonistic bacillus subtilis, first in the biological control as the plant is the antagonistic bacillus subtilis in the Canada, and another is the 1932 William the suggested the use of trichoderma as a biological agent. Waxman in the just like the broad spectrum discovered the broad spectrum antibiotics streptomycin. Nowadays streptomycin is banned where the नॉट बैंड जहाँ पे ये स्ट्रेप्टोमाइसिन का सब्स्टिट्यूशन है वहाँ पे उसको बैंड कर दिया गया, और अगर सब्स्टिट्यूशन नहीं है जिस फील्ड में उस चीज में आप स्ट्रेप्टोमाइसिन का यूज कर सकते हैं. These are all sequencing of the digital development of the evolution of the IPM and integrated disease management. Green revolution has attained self-sufficiency in the food through introduction of the hybrid and high-yielding variety. After the green revolution, the high-yielding variety and high fertilizer and user, and the demand for the more and the demand more of the input in the form of the fertilizer, especially in organic fertilizer, which in turn of the attracted to the more pest and diseases. This necessity intensive control the measure and the damage to the cow, the crops. The continuous use of these chemical pesticides lead to the pest resistance, residual ecological imbalance by killing the predator parasites. These are also imbalance of the integrated and another macroorganism, and the importance of the integrated approach to the integrated disease management when the concept of the concept was developed in international study in 1984. The another is a clearly that the publication in 1963 of the Rachel Carson, the book written by Rachel Carson, the name of the book is Silent Asleep, made the public aware that insecticides such as DDT and the detrimental effect on the clear environmental condition. Why you move to the IDM? Because over the past few decades, the application of pesticide was dominant from the disease control in the developed and increasingly in the developing country. In the late 1960s, the movement of the developed more environmentally being the crop protection method again, and the crop scouting to determine the aspirate schedule. It was the first real step toward the IPM approach. The IDM integrated disease management gives the preference to other control methods, and just like host plant resistance, pest, cultural practices, biological control, cross protection, genetically engineering approaches. They are also genetically engineering biotechnological approach. They are also approaches used for the integrated disease management. In the century 25, the agriculture sustainability is a very powerful word, and they can use it and also maintain the become one of the modern agriculture and the enormous and the non-invasive method, the control of the crop disease such as a pathogen, free seeds, disease resistance, crop rotation, plant extract, organic amendments, biological control are considered less harmful than synthetic chemical pesticides. No any single method can provide the satisfactory control of the crop diseases. The integration of the all effective and eco-friendly measure in the accordance with the dynamic of the ecosystem management will be the best strategy for efficient control of the crop disease. The adoption of the integrated disease management against the disease encountered in the crop is the utmost importance because the dependency on the chemical for the management of the various disease is a great health hazard to the consumer. The use of the integrated approach in the plant disease management is the cost-effective, renewable, eco-friendly, non-toxic to the plant as well as non-target organism. And the beneficial organisms are not destroyed by by using the integrated approach, and that means with the integrated disease management approach. Aim of the integrated disease management, the selected item and the item strategy in the prescription, the promote the natural control and protect the human health, minimize the negative impact to the non-target organism, enhance the general environment. General environment, that means the agro-ecosystem, environmental ecosystem. Also, we most likely to produce the long-term beneficial result, be cost-effective in the short and the long term, be easily and efficiently implemented. Integrated disease management is the concept that from the successfully integrated management and system and developed by the control. The most of the idea is exporting the timely application of the company's trade and the practices. These may be included the site selection and preparation, utilization, resistance, cultivar, the altering the planting practices, modifying the environmental by drainage, irrigation, pruning, thinning, and setting etc., and applying the pesticide if necessary. Monitoring the environmental factors just like temperature, moisture, soil, pH, temperature, the disease forecast, distribute the economic threshold, important the management scheme, and for example, the balancing the fertilizer. The fertilizer is the most important fertilizer application and use of fertilizer. The most important in the enhance and reduce the plant diseases. In case of in case of the we use the highest dose of nitrogen, the increase the susceptibility of the crown gray leaf spot, rice leaf spot, cigarette curly and these also. These measures should be applied in a coordinated, integrated, and harmonized manner to maximize the benefit of the each component. Just like example, the balance the fertilizer and promote the health vigorous plants. The component of the IPM, and they are flowing component of the host plant resistance. It is an inherent host plant resistance. That means the ability of the host to the resistance. He has escaped the infection to the plant pathogen he has stage. That means the host plant resistance. It is an inherent ability of the plant to prevent or restrict the disturbance and subsequent activity of the potential pathogens. Just like a particular host may be the resistance against all races of the pathogen. Let's say horizontal resistance, yeah, may be effective against a few races of the pathogen. The vertical resistance, the control measure by the development of the new variety resistance to diseases against diseases. The breeding of disease resistance is achieved by the usual method, just like introduction, selection, hybridization, mutation, polyploidy, budding, grafting, etc. method are used to the in case of the breeding and the advancement of the selection of the breed. The use of resistance variety is very much welcome by the resource poor farmer because it does not require the additional cost and it is environmental eco-friendly. The principles of the principles of the plant disease management. First of the principle is the avoidance, exclusion, eradication, protection, and immunization. One minute, sir. In case of the avoidance, that means the avoid the proper selection of the geographical area and the selection of the field, the free from the pest, the pest, that means the pathogen, and avoiding sowing dates and choice of the disease escaping and variety. Another is exclusion. Exclusion is the quarantine measures, seed certification, plant disease notification, and prevention of the scale of the disease and the plant, and eradication. That means the use of cultural use of matter to control the plant disease. Just like the cultural practices, physical, biological, and integrated and chemical practices. Another is the resistance, that we have the protection. There is protection and immunization with the host plant resistance. Effect of the cultural practice on the disease control. There are two types of effect: direct and indirect effect. Direct effect, that means reducing the amount of uroculum and reducing the speed of the expansion of the pathogen on the host. And another is the indirect effect by increasing the population of the microbial agent in the soil, which will be helpful in the controlling the pathogens. Cultural practices, the benefit of cultural is the control within the stimulants for the growing environment that favor of the crop over the pathogen. And the cultural practices such as the cultivation techniques, mulching, intercropping, plant density, planting dates, crop rotation, strip farming, time of the harvest, varied crops, crop mixtures, growing, healthy planting, soil solarization method. The soil amendment is just like the organic method, and the fertilizer management, water management, the use of the water management just like drip and sprinkler irrigation have been used singly, and the combination of the tool for the disease management, that is the practices of the culture. Soil solarization, the first of the soil solarization, the covering of the soil with the material, the plastic material, the which rise the surface temperature above the evident, the using the plastic sheet, the soil solarization, the transfer plastic are used for transplant polythene sheet laid over the soil during the period of the high air temperature, and it can reduce, eradicate the variety of pathogen and pest, including the soil-borne fungi, nematodes, and weed also. There are so in regions, the most of the pathogen propagates are unable to withstand temperature of the 50 degrees Celsius for more than a few hours. At longer period, low temperature may be the lethal. The soil temperature rise above the critical threshold level. There actually pathogen propagates killed and escape the infection of the hosts. These are they are quoted with the example: the onion whitewort, that's especially cephalosporin, and the many soil-borne pathogen are controlled by soil solarization by using the seeds. Deep plowing and plowing the land during the hot summer period in the early monsoon period, crop residue exposing the resting spore and the sun thermosensitive pathogen die, the pneumovolt and species. These are the kicker hypa, Sclerosia, rhizomorph, and macrosporidia, and the O spore, chlamydospore. They are deep summer season में deep plowing करते हैं. These are also these spore are survive as a dormant spore, but in case of the summer season plowing, they destroy and to escape. Do not chances the infection of the plant disease in the next semester and the primary infection. They check. Another matter is the flooding. Flooding matter is very much matter. Thus, in some cases of the pathogen, just like fog, featuring oxysporin cuvans is a highly destructive pathogen causing the banana wilt disease. The tier 4 cases are reported in some country and also including India. The field that summer will hit resembles soil disease infection, long-term summer soil flooding with without paddy culture is found to be the decreased population of the soil-borne pathogen. Another is sanitation practice, use of sanitation to remove the infected plant virus from the field, just like hawk leaves, microspora. The hawk leaf, that means the fusion of oxyspora propagates are survive on the soil, and infected plant material of the banana, they can destroy sanitation. And another is they avoid the soil movement. Another is no transporting of the soil, because the infected soil are transported to another place. They also infect the soil, the whole field. बहुत सारे केस में ऐसा होता है कि फार्मर से कहीं कहीं से सॉइल लेकर के अपने खेतों में वो डालते हैं ताकि वहाँ का सॉइल का जो न्यूट्रिएंट कंसंट्रेशन होता है या सॉइल का जो टेम्परेचर होता है सब मेंटेन करने के लिए। बट ये गलत मेथड है क्योंकि वहाँ पे जो भी पैथोजेंस हो सकते हैं वो कंटामिनेट कर सकते हैं अच्छी सॉइल को भी। दे डू नॉट appropriate method for the management of the soil-borne diseases. Crop rotation is a very important practice, especially for soil-borne disease control. For many soil-borne disease, at least 3-year rotation using the non-host crop greatly reduce the pathogen population, just like the practice of Phytophthora blight of the pepper and fusarium wilt of the watermelon. But longer rotation period, 5 to 7 years, are required for the complete. Do not completely destroy the pathogen, but a maximum percent of the pathogen is eliminate from the field. In case of the potato cyst nematode, these plants are contain the nematicidal properties. Also, they can yield the field. Soil-borne pathogen, just like Macrophylla fasciolina, can be controlled by the growing smoothing crop and fusarium wilt in the red grass by sorghum. In case of the fog, in case of the fusarium oxysporum, they can eat the marigold and rice cultivar. The some previously recently research paper published by a BHU scientist. The fog is controlled by the fog is rotational by the rice crop because the rice is in the field of the rice. The always maintain the some degree centimeter of the water in the field, so can eliminate the pathogen because the starvation of the oxygen and they do not survive in the field. Weed control is also important because some weed are alternate host of the pathogens, and they infect as a primary infection is from the host. Another is the excessive handling of the pathogens, and the soil aeration and drying can be enhanced the intercropping composted organic amendment in the soil. The pathogen inoculum can be reduced by the removing the plant material infected and healthy after the harvest. The following physical methods, the physical and mechanical measures. The mechanical can control kill a pathogen directly, yeah, make environmental unstable for it. This is a common method that collect and destroy the disease infected plant parts. Another is soil sterilization at 50 to 60 degrees Celsius, about the 30 minutes, kill the soil-borne pathogens. There are so many, the soil-borne disease like lose amount of the heat. They are 52 degrees Celsius or the 11 minutes. The treatment at the leaf scale 50 degrees Celsius for 2 to 3 hours, red rot 50 degrees Celsius for 8 hours, red extending of the sugar cane 50 degrees Celsius for the 3 hours, and black rot of the crucible 50 degrees Celsius for the 20 to 30 minutes etc. These all are the method for the physical method, the seed treatment. 1 minute, sir. Again, 1 minute. Hot air treatment is given to remove the excess of the moisture from the plant organ and protect them from the fungal and bacterial attack. The several virus infected dormant plants are treated by the hot air treatment at a temperature ranging from the 35 to 40 degrees Celsius for 8 hours. 35 to 40 degrees Celsius for the 8 hours that destroy the several bacterial pathogens. And the refrigeration, low temperature treatment is the most common method used for prevent the post-harvest disease of pathogens. Another is the biological control. The previously slide are the most discussed the biological control. That means success using the microorganism against the pathogen started with the control from agrobacterium, radiobacterium, KST94 and the K9080. The pythium, they control they defend against the Pythia, Rhizoctonia with the Trichoderma argine. Another is the cross protection. Cross protection is also one of the modern biocontrol method for the disease management. In this method, the mild state of the virus, microorganism, pathogen, is inoculated in the host. These provide the protection of the host plant from those viruses and microorganisms which may be cause the more severe damage in the host state. Just like example, the papaya spot disease and citrus diseases. One of the limitation of the using the biocontrol agent is their inability to survive in the certain field condition. That means with the host specificity and limitation to the exploration and the lack of handling. Each of botanicals, they are previously research report I described in the age of botanical is just like lemongrass, a number of botanicals are also tested against the fungal disease like C. coli. The some commercially used the botanicals, neem, garlic, the tobaccos, ginger, thyme. They are also reported as the lemongrass. They are also botanically pesticide are used nowadays, and substitution of the highly toxic chemicals. They can also enhance they stimulate the growth, but they are also defend against the plant pathogens and bacteria and fungus. Plant quarantine regulation. The plant quarantine is a legally forced restriction on the movement of the disease plant or material from one place to another place, just like bacteria from the virus that cause the disease in the plant. When the plant pathogen are introduced, when the plant pathogen are introduced in area which did not previously exist, they are likely to cause much more testophoric epidemics than do the existing pathogen. And some of the worst plant disease epidemics that have occurred through the world, for example, of the down mildew of the grapes in the Europe, and another is the bacterial cancer of citrus, and another is the chestnut blight. The operation of the quarantine regulation are also important disease. It is extremely difficult to predict accurately because whether the exotic organism will become the established, and the once established become the economically important. And the purpose and the intent of the Quarantine Act in the country to prevent the introduction of the insect, fungus, bacteria, virus from a country. Further, the significance of plant quarantine is the increase in the view, globalization, and liberalization of the international trade of the plants and plant material in the wake of the century of photosanitary certificates and agreement with the World Trade Organization. The photosanitary certificates of agricultural commodity being the export also undertaken through the scheme as per the International Plant Protection Convention IPPC 1951. Another way the chemical control that is two types of chemical, either non-systematic and systematic chemicals, and the contact nature of the chemicals. Factor influence the disease management strategy. There are many strategies available to manage and control the plant disease. However, just like before, the most of the appropriate strategy can be recommended. The several practical considerations must be taken in account. These including economic advantage of the control, the facility of the control available, and the disease in the perspective. The economic advantage of the control. Determine the economic advantage control, necessarily the economic return from the crop if no action is taken, and another is the likely economic return of if the disease is controlled by the particular method, as well as the cost of the various control options, include the cost of the material, labor cost, and any damage likely to the caused by the treatments. Having estimated the various costs of the benefit likely to result from the various control options, the economic advantage. Economic advantage of the disease control, the expected return if disease is controlled, expected return if disease is left, the uncontrolled plus cost of the control treatments. Fragility of the biological control, the fragility of the cultural control, that even through the cost benefit analysis might indicate the economic advantage if the particular control option is implemented. One might decide not to be recommended any control option. There is no point in the recommended. The option available, any control recommendation should be always taken into the account of the STC product in that question. The option available to grower must be considered. And the disease in respective, the threat of a dangerous disease to the industry as a whole, negative vegetation will influence the control strategy yield. If the disease is caused by the newly introduced parasite not previously found in the area, and if the disease constitutes a serious threat of an important industry, the control strategy, regardless of cost, might be utilized. Thank you, sir, and thank you, dear students and participants. Manoj sir.
Hi, yes.
Yes, sir. I will conclude the topic, sir. But a very low number of participants attend this seminar, sir, because I don't know. I हो सकता है कि मैं अच्छे से उनको समझा नहीं पाया
No, since the midterm examinations are going on.
Oh, okay, sir.
One of the reason that students are not there. Okay, so then let's if there are no any questions, we will stop here then. So on behalf of postgraduate institute of Rahuri, I am very much thankful to